Excuse me, can you hear me?
Yes.
So actually, I have two accounts, and I joined using two accounts. And yeah, full name account is my main account.
We can hear you, Ishihara-san. We can hear you.
Okay. Mm-hmm.
Also, you
Okay.
were
Yeah. No problem. No
PPT.
problem. Okay.
Okay. See, you're fine.
No problem. Okay.
Is Saito-san join?
Saito-san.
I will give him a call.
Hello, Ishihara-san, will start now?
Yeah, sure.
Good afternoon, everyone, and welcome for the second session of the day. And today joining us is Hiroya Ishihara-san from Olympus, and it's my great pleasure to introduce him. Ishihara-san is an application scientist at Olympus. At the Tokyo University of Science, he studied factors involved in plant regeneration using omics and microscopy. Currently, he is working on a wide range of projects from basic research to products and sales strategy. And before we get started, there are a couple of instructions for the participants. Please keep your audio and video off at all times until you are instructed to do so. And if you have any questions, you can just type in the chat box and they'll be directed to the speaker at the end of the session. I think that's it. We can get started, Ishihara-san.
Okay, let's start. Hello, everyone. Thank you very much all here for coming today, and it's good to see you all here. And yeah, let me introduce myself just a little. And my name is Hiroya Ishihara. I'm an application scientist at Olympus, Tokyo. Yeah, as he said before, I was studying epigenetic factors involved in plant regeneration using transcriptome and microscope. And in 2019, my paper was finally published in Nature Communications. And at that time, the microscope was my trusted partner. Therefore, I joined Olympus. I joined Olympus to make life science more exciting with microscopes. And today, I'd like to talk about 3D segmentation-based analysis. And I'd like to talk not only about 3D segmentation, but also about the samples and imaging. It is also important topics for 3D cell analysis. After this session, you will immediately want to try to using 3D cell analysis. Try to do 3D cell analysis. Okay, let's start my presentation. And here is my agenda in this presentation. First, sorry, okay. First, I will introduce the overviews of the 3D cell, 3D samples, and 3D cell analysis. And after that, I'd like to talk about sample preparation and microscopic observation, and 3D segmentation, which are important techniques for 3D analysis. In the 3D segmentation part, I will talk about our product, the Noviside's 3D cell analysis software. Then I will introduce application cases of analyzing 3D cell model. Here, I will introduce examples of antibody drug and immune cell therapy evaluation using patient-derived organoids, and high-throughput drug screening using co-culture spheroids. Finally, I will show you a demonstration video with my explanation. The 3D analysis part, the demonstration part, is based on our software, but I hope you can refer to the concept and the way of thinking. Please enjoy my presentation. Okay, first I will introduce the overview of 3D cell analysis. So a variety of models are used in life science research, there are a variety of models ranging from cell monolayer to individual level. And they need to be selected according to your purpose, right? And among them, the organoids and spheroid are very well-balanced models in terms of biomimicry and easy to use. And what kind of model do you usually use? So if possible, please let me know in the chat box what kind of samples you're using. So if you can possible, please type and send me. Okay. Oh, thank you so much, East. Okay. Okay. And the organoids and spheroid are widely used to model and study tissue development and cancer research and regeneration research. Sorry. Okay. Tissue development and cancer research and regeneration research. For example, the organoids are used to identify the factors required for stem cell to differentiate into specific lineages, such like the lung and heart, something like that, and the molecular mechanism of cell fate programs. And organoids also enable knockout studies of essential genes that are not feasible in vivo due to the lethality of the embryo. And the pluripotent stem cells derived organoids are a very important model system for developmental biology. For example, analysis of human and non-human brain organoids at the single level has revealed human-specific features of neurogenic processes. And next, I want to introduce example of research using organoids. So this paper is an example of use of organoids as a model for developmental research. They reproduce the Neanderthal brain organoids, our ancestor, and using CRISPR-Cas9 system, and compare them with human brain organoids and Neanderthal brain organoids. Then the image-based analysis of proliferation and surface complexity show that there were differences between Neanderthal brain organoids and human brain organoids. Although there are many questions that remain, like that they have not been able to reproduce an exact Neanderthal brain. But it is very interesting study. So organoids and spheroids are very interesting sample in terms of the biomimicry and easy to use. So yeah, you may have an idea of samples so you want to use and things you want to do, but the many models like organoids and spheroids are three-dimensional. So how do you analyze and evaluate those 3D models? Do you have any idea? Or so therefore, we proposed a method of imaging transparent samples with a confocal microscope and analyzing it in 3D using appropriate software. So actually, this is a session on 3D segmentation, but the sample preparation and microscope are also important techniques, and I will talk about them step by step, okay? So the reason why it need to be analyzed in three dimensions is because 3D model have a problem with heterogeneity. So by imaging the whole model and analyzing all cells, and we can quantify the signal against the internal control. And the confocal microscope and two-photon microscope are a good match for imaging the entire cancer model in terms of speed and image quality. For larger samples, tissue clearing techniques can be used to acquire a whole image. And so the software is also needed to properly analyze the acquired images. And we have recently developed a three-dimensional cell analysis software called Novisite. Yeah, I want to introduce our software later. And let's start at sample preparation. So even the most powerful microscope cannot be used for observation if the laser cannot reach the sample, right? And biological tissues are, so biological tissues are composed of various molecules, and the refractive index of the entire sample is not constant, right? Resulting the scattering the laser beam. So the clearing region make the entire sample uniform in refractive index, thereby eliminating the scattering, right? And make it transparent. So we often use scale view S4 as a clearing region, which are a water-soluble clearing region. And scale view S4 can easily make entire sample transparent. And the silicon immersion lens is good match for this clearing region. And scale view S4 can be easily made transparent by simply immersing the sample after fixation and staining. And if you are using 3D cell models like organoid and spheroid, you can try it out. It is very easy to use. And next, I will talk about microscopic observation and microscopic observation for 3D cell analysis. There are many microscopes used for 3D cell analysis, 3D imaging, and it is necessary to select the appropriate modality according to your purpose. Right? Actually, the confocal microscope system is the well-balanced and versatile systems. The other three types have their own special points. For example, the two-photon microscope are suitable for deep imaging. And the spinning disk confocal microscope are capable of high speed and high resolution imaging. And the light seed microscope are capable of high speed observation of a wide range, a wide area and depths. But they have low resolution and sample limitation. So you need to choose the imaging modality according to your purpose, right? And so as I mentioned in the clearing region, the refractive index matching between the sample and objective lens is very important. So if you are mounting with a high refractive index region, like glycerol or clearing region, like scale view S4, the silicon immersion objective lenses are recommended. And so if you use large samples, we recommend a two-photon microscope that are deep observation using a long wavelength laser. It enables bright observation in the deep area. And because the laser beam not reach deep area, so since the laser beam doesn't reach the deep area, it is necessary to adjust the laser intensity and detector sensitivity according to the depth, like this. So FV3000, our confocal microscope systems, has bright depth function. So this function enable imaging with the appropriate setting depend on the depth. And I'm going to say this again and again because it's very important. Please keep this in your mind, at least in my presentation. So there is an inseparable relationship between the clearing region and objective lenses. Okay, so because the refractive index of scale view S4 is 1.49, the silicon immersion objective lens is the best, the most compatible, and can acquire clearing images to the depths like this. It is very important. So if you use the clearing region, so you should use the silicon immersion lens. And the analysis result depend on the how detailed the image is taken also in the Z direction. For example, sorry. In this example, the 226 images were taken in 1 micrometer increments and 23 images in 10 micrometer increments. As you can see, it affects object recognition like this. So the more detailed, the more accurate. But of course, it takes more time. It depends on your purpose and your target of analysis. Okay. Okay, let's finally talk about the main topic, 3D segmentation. Here, I will talk about our 3D cell analysis software, Novisight, as an example. So we developed the Novisight, a 3D high contents analysis software. Let me explain the 3 features of Novisight. First, the Novisight offers true 3D analysis. And second, the Novisight is suitable for analyzing multiple images from micron plate, from material plate. And third, on the Novisight, the data and images are connected. Everything is interactive. So this is the features of Novisight. And so true 3D cell analysis is a method of spatially analyzing 3D images. In some cases, 2D section images or the projection images are used to analyze 3D images. In contrast, the Novisight spatially recognized and analyzes signals in the 3D images as a voxel, like this. And these are all more information to be obtained and complex model to be analyzed in detail. By obtaining all information in the 3D images, it is possible to accurately analyze 3D models. But this is a basic procedure for 3D cell analysis. First, pre-processing the image to detect object, and then do the detection, the object detection, and then the parameters in the object are measured, and the parameters are used to classify the desired object and get the final data as a table or heat map. I will explain the step by step. So pre-processing make the foreign object recognition easier. And the background processing is mainly performed here, like this. This is the background, the mechanism of background processing. For background processing, we use the rolling ball process as an algorithm. Just a set radius of rolling ball, it will estimate the background intensity and clear the image like this. So it is very useful and easy processing. And next, it's about detection, object recognition. The bias recognition algorithms are available, like for nuclei and whole sphere and the organelles, something like that. So we can detect the various object. And this is a mechanism of 3D segmentation. First, the center of the objective is identified based on the intensity. And then expand the area to determine the range of the object, like this. Yeah, but it is very simple mechanism. And after detection, the additional filters and the processing can be performed for cytoplasm and intracellular analysis, like this. So this is the it's about analysis for cytoplasm, and this is for intracellular. And the set the parameters you want to measure for the detected object. Here you can select parameters related to counting, morphology, and intensity, and location, and tracking. So we can choose that from these parameters. And use the measured parameters to perform classification. Extract the desired object using histogram and scatter plot. For example, the small object with high intensity, or the object with large surface area and the small volume, something like that. So you can set it freely, and you can extract anything you want. So using measured parameter. Yeah, it is classification. And finally, the display measurement and population result for extracted object in heat map and table. Of course, you can also display the result in a viewer like this. And next, I introduced an application cases of analyzing 3D cell models. First, let's talk about the application of patient-derived organoids. So this application was developed in collaboration with Fukushima Medical University in Japan. The first application is spatial analysis of fluorescence-labeled antibody drugs in FPDO, the Fukushima patient-derived organoids. The patient-derived lung cancer organoids are treated with antibody drug conjugated with high fluorescence dye, and binding region are quantified. As a marker, we are using Herceptin, which targets HER2, and Erbitax, which targets EGFR. Okay. And after imaging, we used Novisight to measure binding region of antibody drugs. In this process, we not only measure the whole organoids, but also divided the organoids into the central and edge region, like this, and measure the binding region in each region, like this. The observed images show the Herceptin was irregularly bound to RLU and 21, like this, while the Orbitax was mostly bound to the surface of RLU and 21, like this. By quantifying the volume of positive regions, we are able to calculate the positive volume, like this, the positive volume rate of antibody drugs, like this. And then by analyzing the organoid, the edge, the outside, and inside, the separately, we are able to quantitatively show that Orbitax is strongly bound to the edge of organoids, like this. So we can quantify the antibody drugs, the volume of antibody drugs using Novisight software in 3D. And second application is three-dimensional cytotoxicity analysis of immune cell in patient-derived organoids. A natural killer cell are added to patient-derived lung cancer organoids, and the number of apoptotic cells in organoids is counted. So we use the NucView 5300 as an apoptotic marker. And the imaging results show that the natural killer cells were treated for 3 hours, which induced apoptosis, like this. And then, so we want to quantify the cell viability rate, the apoptotic cell rate, due to immune cell treatment. So to count apoptotic cells, we first recognize all nuclei, like this, and then the recognized object are classified into on a scatter plot. In this case, we use the NucView intensity to define the apoptotic positive cell and negative cell, like this. The high intensity, the high total intensity and high mean intensity object is apoptosis positive cell. After that, we count the number of apoptosis positive cell, like this. And so as a result, so we are able to quantitatively show the effect of natural killer cell in patient-derived organoids. And next, I'll introduce the high-throughput 3D analysis using co-culture spheroids. So co-culture spheroids on 384 well plate, 384 well plate format are imaged and the number of cells in each is counted to evaluate the effect of drug. The spheroid used in this study is the co-culture spheroid of colorectal cancer cell and fibroblast, so which express EZFP and RFP in each nuclei separately, respectively. Sorry. So the cancer cell expressed EGFP and the fibroblast expressed RFP. And the spheroid in this study, so we will quantify the effect on each cell when treated with the four different drugs, right? So these drugs. In order to acquire high-resolution images at the high speed for use in this in the analysis, so this time we optimized the correction color of the objective lenses and the step size in Z direction and the averaging times for fast scan. First, let's talk about the correction color of the objective lens. So when using an objective lens, which with a correction color, be sure to adjust the correction color. So the appropriate position depend on the objective lenses and samples. In this condition, I was able to acquire the deepest image at the 0.17 position like this. It is much better for this analysis. And then it's about these steps. In general, the finer the step size, the higher the recognition accuracy, but it takes a long time. So in our study, so for general nuclear recognition, the step size should be set at 3 micrometer or smaller. And next, it's about averaging. The image quality of high-speed, the resonance scan, is determined by the number of averaging. So in this case, the object recognition was possible without averaging. It is very fast. As a result of imaging under optimized condition, high-resolution images of 252 samples could be acquired within 1 hour. The 14 seconds per samples. And then we start the analysis. And the drug effect is evaluated by counting number of each cell after drug treatment. First, after all nuclei are recognized, and then they are classified into 2 cell types using GFP and RFP intensity. Then the number of cells in each is counted. So this is the result of object recognition. After object recognition, the object were classified into two types of cells. the cancer cell and fibroblast. In Novicite, so you can easily check the result of the individual well using heat map function like this. So this is a heat map of each of the cell count for each well. The two end group, this and this, are the control group, and the drug concentration is higher toward the light. So this is a high concentration area, and this is a low concentration area. The high concentration of the drug showed a decrease in number of cells, and Novicite can quantify the increase or decrease in number of each cell in the co-culture spheroid. And then this graph shows the change in the number of each cell at each concentration of treated the sunitinib. The sunitinib was found to be more effective against HCT cells, the cancer cell. Okay, so this is my presentation part. So if you have any question, I want to answer them.
Ishihara-san, there was a question. Can you please explain the rolling ball concept?
Rolling ball concept. Okay. Rolling ball concept, please wait. Oh, sorry, please wait. Ball concept. Okay, can you see that? Okay, this is the mechanism of background processing. So, should I say? So we set the radius like this, and sorry. So this is the actual intensity in each pixel, and so we made a 2D curve like this, and then we set the radius like this. So if you set the small radius like this, so this the small circle are a lot of small circle are made it here, and so you can estimate the smooth background. So how should I say? So the large radius if you make the large circle, it is a rough estimate, the background estimate roughly, and if you set the short radius, you can estimate very sharp. Is it okay? So actually, I'm a biologist. Sorry, I'm not sure about the detailed mechanism and algorithm.
Maybe you can just tell that, like, if the radius is bigger, what is the effect on the background correction? if the radius is smaller, what is the effect on the background correction or background
So
processing?
yeah. So if you set the big radius, so the how should I say?
Background
So
correction is poor.
correct.
Background
So
correction
if
is poor. If you make it smaller,
mm,
it'll be excessive background correction. So like it might
mhm.
even like take out your signal strength also. So you have to do it carefully. If the
Re-
background is too much, then the radius should be bigger. If the background is less, then the radius should be bigger. If the background is less, then the radius should be higher.
as if you want to background process strictly, so you should set the small radius to make a sharp images.
Are you doing it in the demonstration here?
Yeah. Oh yes. Yes. I can show you later. Yeah.
Okay, fine. I think that.
Mm.
Alice, does it answer your question, or so maybe like I think it will be shown at the end. At the end.
Mhm. Oh, okay, sir. Thank you. Anything else? Okay. Yeah, you're welcome. Okay. Okay, let's go to the demonstration part. So please wait. Prepare my demonstration.
Hello, Shara sir.
Hello?
Before the hello, can you hear
Mhm.
me?
Yeah,
Before
sure.
the demonstration, would you like to take a 10-15 minute break?
Oh, yes. Okay, yeah.
Okay, then we'll take a 15-minute break and then we'll meet back at 3 for the
Okay,
demonstration.
okay. Okay, see you later.
Ishihara-san?
Hi, I'm here.
We can get started.
Okay, let's start the demonstration. Oh, okay. Yeah, I want to show you the demonstration video. And in this session, I talk about our 3D cell analysis software. We call it Novicite. Rather than detailed operation of Novicite, I'd like to tell you how to think about it, the way of thinking, the concept for analysis. Okay, I will show you the two cases: the mitotic cell analysis and the autophagic cell analysis. Okay, let's start about mitotic cell analysis. Okay. So, okay, let's start mitotic cell analysis. This application identify the number of mitotic cells in spheroid. And spheroids of 8029 cells are prepared and staying with the DAPI and made transparent using scale view S4. Oh, sorry. And we use the confocal microscope with silicon objective lens. And this is the strategy. In order to do the analysis, we need to define the mitotic cells. What are characteristics of mitotic cells? So to determine our mitotic cell from image, we use its intensity and shape. Okay. In other words, it is the small nucleus with a high maximum intensity. Okay. The first, all nuclei are recognized, and then the morphological and intensity information is used to extract the small nuclei with high intensity, like this. And yeah, we define this nucleus as the nucleus of a mitotic cell. Okay. Okay, let's start my video. So this is the GUI in Novicite. So this is a 3D cell analysis software. And first, we will see the images and before analysis also. And so we want to count the mitotic cells. The mitotic cells are our target. So we can see the three-side view, and we can also see the volume using volume viewer like this. And we want to count the mitotic cells. First, so it is object recognition. So in this case, so we skip the image processing. So we don't need image processing. So because this is a champion data, this is a high resolution and low background intensity, so we don't need to use the image processing. And then so we want to recognize the object, the nuclei. So we will do the object setting. So we use the sphere algorithm and nuclear L algorithm for this channel. So sphere algorithm recognize the the sphere algorithm recognize the entire spheroids, and the nuclear L recognize the individual nucleus. Okay, we will recognize the two object. And first, we will set the we will do the setting for sphere module. So we use the O2 function as a threshold. It is the threshold O2 is the automatically binarization, and actually, so we also do the we also set the threshold, the manual binarization. So in this case, we use a threshold O2, the automatic binarization. And we first recognize the whole spheroid. And we will check the preview. So this is a recognized result. So we can also show the display outline like this. And we can also see the result on the 3D side view. So we can see the XY axis and YZ axis and XZ axis. So this is the whole the recognition for the whole spheroid. And then we will set the nuclear L algorithm. So nuclear L algorithm is very simple. So there are three parameters to recognition to recognize the nuclei, the cell size and background intensity, and the XY ratio. So in this case, we in almost case we use the cell size and background intensity. So we don't use that the XY ratio. It is a special case. The cell size is a set diameter of the nucleus, and background intensity is a set threshold. And the 0 means the automatically thresholding, auto binarization. First, we check the preview. As the cell size is 10 micrometer, so we will check the recognized result. And display outline. So we want to see the more detail. We will set the limit preview area that we focus on this area. Okay, this area. And click on preview again. and we will focus on this area. And we can see that this area, and so I think that we need more separation, so that the two cells, two nucleus, as counted is counted as the one nucleus. So we need more separation. So we should change the condition, like the set cell size 10 micrometer to 8 micrometer to recognize the small nuclei. And we will check again. And yeah, it is good separation. It is better. And we will check we will do the final check in whole spheroid. Mhm. And you check the whole spheroid, the recognized result in whole spheroid, and check in 3D side view, and you can see the recognized result like this. Oh, it's better. Sorry. And then, so next, object ring setting. So basically, in Novicide, the object that are contained within the main object will be detected. So this means the parent of analysis is the main object, and the object one. So sorry, the main object is the whole sphere, and object one is nuclei, like this. The main object is sphere. The object one is nuclei. The only object contained in the main object or analyzed in Novicide. Okay. So if we want to set if we want to change this relationship, so you can set the object ring setting, but the specific adjustment are not necessary this time. It is the simple case. In next case, I can introduce this function later. And next, the parameter setting. So we can set the parameter in each object, and we have a lot of parameter, like it's about intensity and morphology and position, something like that. And in this case, we choose the measure axis size. The measure axis size means the wrong part of nuclei for object one. Object one is a nucleus. Okay. We set the measure axis size for nucleus. And then, so we also use the maximum intensity for channel 2 in object 1. And we set this parameter. And next, run analysis. So the analysis setting are complete. And then, so we will do the running analysis and the finish recognition and show results in this area. And next, we will do the classification to define the mitotic cells. And we can use the graph with the using the rotative type of axis, and we will set the x-axis, the major axis size as x-axis, the max intensity as a y-axis, okay? And the graph will be created. The x-axis is the major axis size, and y-axis is the maximum intensity for channel 2. And in Novi site, if you click on plot on the graph, that display the image of corresponding object, like like this, and yeah, you can check. This, the corresponding object. You can see the corresponding object, and you can check the boundaries between mitotic cells and normal cells. Click and check, and click and check, and you can see you can find the boundaries using this function. And as a result of the find try, this region is the mitotic cells, and we can create the region to classification. And we can see the region. We can see the images contained in this region. We can use the object gallery function. So this is the classification results. But there are also this is the mitotic cells, but these are normal cells, I think. And if you click the thumbnail you want to check, you can see the viewer and graph, and you can know which objects are normal cells, and which plot is normal cells and which plot is mitotic cells. And you can modify the region. You can check and you can modify the region, and check the result in the gallery view again. And yeah, there is also the normal cells. This is normal cells, and we need to modify the region and check. Yeah, it's get better, I think. Yeah, this is the classification. So we use the graph and measured parameter. In this case, we use the intensity and morphology. the small nucleus and high intensity. Okay, we use that. And next, the gating. So gating means the classification step. We made the region for the other mitotic cells, and next we will define the mitotic cells using this region, and right click on the region and click on gate, and then gate is created, and we can adjust the gate. So R01 means this region, the small nuclei and with high intensity. So this is a gate for mitotic cells. The definition is R01. R01 is this region, and we need to create the normal cells, the gate for normal cells. So we create the new gate, and definition is the not R01. So change the definition using Boolean expression. The Boolean expression is and or not, something like that. The normal cells is not R01, like these. Okay, so this is R01 mitotic cells, and these are not R01, the normal cell. Okay, we can create the gate to define the object using Boolean expression. Okay. And we can change the color, gate color, to show the result. And then we want to compare the mitotic cell, the images of mitotic cells and normal cells side by side. And we display each gate on graph. The left side is mitotic cells, and right panel is the normal cells. And we show the region gallery for each gate. So this is the region gallery for mitotic cells. And right panel is the region gallery for normal cells. And check each gate. And left is mitotic, and right is normal. Yeah, it is. I think it is a good separation. And this is a gating. So gating is a definition. Okay, and then we will see. We want to see the result as a number. And upside display the result table and the result heat map. In this case, we use a single images, single sample. We don't need to use we don't need see the result heat map. So we will see the result table. In the measurement result, you can see the measurement result in this. And you can see the population result and counting result in this tab. In this case, it's about 7,000 nucleus are in the spheroid, and 78 cells are mitotic cells. And we can see the display populations. And we can change the display to the percentage, and we can check the result. In this case, about 1% of the cells were mitotic cells. And if you prepare the other sample, like arrest the cell cycle, like I forgot the name. If you treated the drugs to arrest the cell cycle, the mitotic cell ratio will be increased, I think. So you can quantify the mitotic cell ratio. And then, so if you click on download table, so you can download this result as a CSV file, and you can make a graph on Excel or something like that. And next, the view result on the viewer. So you can show the gate information in viewer. So, okay. Can you see that? The red square. The red square is a gate. The object on the red square is defined as mitotic cells. The red square. And you can also see the volume viewer. And this is a volume viewer, and turn on the objective one. Sorry, click on gate. The display displays a gated object as mitotic cells. These red objects are mitotic cells. Okay, this is the first demonstration. It's about the mitotic cell analysis. So before go to the next case, if you have any question, I want to answer them. Do you have any questions or any comment?
There are no questions in the chat box. Participants, if you have any questions, you can just unmute yourself and ask. There's a question from Kiran. He's asking, is there a trial version?
So it is not so.
We can demonstrate it. We can demonstrate it, and then later on, yeah. So like, please, Kiran, you can let me know, so I can just put my email ID in chat box. So we can try and demonstrate it online, or like if you are somewhere nearby. So we are probably putting this machine in IISc and JNCASR for demonstration. If you belong to any of these institutes, or if you are there in Bangalore, you can bring your sample and then like you can just try it. Later on, we can decide what has to be done.
Yeah. So if we want to use the Novi site, so please contact to Ganesh. Yeah, thank you so much. And we also can discuss about your sample to analyze in Novi site. Yeah.
another question from Alice. Can you show us how to determine protein intensity in the cells?
How to determine protein intensity in the cell? So you mean the protein intensity is the fluorescence protein like G. So if you want to know about the intensity for EGFB in each cell, so first we recognize the nuclei. So you want to know in the cells or nuclei? In the whole cells? In the cell, okay. So actually, the 3D segmentation in so if you want to divide in each cell, it is difficult. But yeah, it is difficult, but we can try it. And if you so the boundary, we couldn't find the boundary of in each cell. So we need to use the nuclei intensity. And so actually, so that next case use this technique that I want to show this case. So in this case, so we have we have a signal in we have a signal on nucleus, and we can expand the region of nucleus to like a cell, the boundary of cell, and we can extract the parameter as a fluorescence protein intensity. So yeah, it is difficult, but we can do that, I think. But it is depend on the sample. But it is yeah, we can try it. Is it okay? Sorry. Yeah, it is a difficult case. So the analysis about the nucleus is easy, but the analysis for the cytoplasm is the more difficult, I think. Yeah, thank you so much. Okay. Anything else?
That's all, sir.
Okay. Okay, let's move to the next case. It is a autophagic cell analysis. So okay. So in order to do the analysis, so we need to define sorry. So this okay. So this application count the number of autophagic positive cells in sorry. I want to count the autophagic positive cell in the spheroids. Okay, and spheroids are prepared from the HeLa cells expressing LC3 EGFP, and treated with the Hekist phosphonuclear staining and chloroquine. So chloroquine induced autophagy, do you know? And we prepared the two samples, the control and chloroquine 100 microgram per milliliter, right? Okay. And we used a confocal microscope with a silicone objective lens. And so in order to do the analysis, so we need to define the autophagic cells. Okay. To determine autophagic cells from image, we use the number of LC3 puncta, LC3 spot in each cell. So in this case, a cell that contains more than 3 puncta, more than 3 spot, is defined as autophagic cells. So this number is based on the paper. In that paper, the threshold of LC3 puncta by chloroquine treatment is set 3. So the analysis is performed starting from nucleus. But it is a difficult case, I think. So yeah, this analysis is performed from starting from the nucleus detection, and but since the LC3 localized in cytoplasm, like this red dot. So we need to be more creative, okay? And so we need to use the object mask function. In first, it first recognized all nuclei and LC3 spot. Then they used object mask function to extend the nucleus area, like cytoplasm. Okay, and in this way, the nucleus will be linked to recognized LC3 puncture in their cytoplasm. Okay, the nucleus and LC3 puncture are linked. Okay, and then the cell that contain more than 3 puncta LC3 spot in the cytoplasm are extracted and defined as a autophagy positive cell, like this. And we count these cells. Okay. Okay, let's start the demonstration. Yeah, we prepare the two images. The right image is a control, and left image is control, and right image is in the treated chloroquine autophagy inducer. And we can also see the 3-side panel like this. And we can also see the volume to see 3D images like this. And next, in this case, we will do the image processing to detect the LC3 spot. Because LC3 spot is very small particle. Oh, it is very... Yeah, in this case, we use the background module. So as I mentioned before, to detect the LC3 spot. And sorry, we select the the channel 2. The channel 2 is LC3 EGFP, the green signal, as the target channel. And we will adjust the background. So as I mentioned before, background has the single parameters radius. Yeah, I want to explain this background processing and the effect of the radius. Sorry. So this is a 50 pixels as a radius. So yeah, this is the result. And so we will change the radius. So the intensity of background is still high. So we want to more reduce the background. And it's very sorry. And we change the radius from 50 to 2 pixels. This is very small radius. And do the preview. It is more strict. The background intensity was suppressed. And we can detect the LC3 puncture as a spot or the puncture. Yeah, we can detect the LC3 signals as a spot or puncture. Okay, so we want to suppress the background strictly. Okay, so we set the 2 pixel as a radius. So this is a background processing. Okay. And after image processing, oh yeah. And after the image processing, we will do the object recognition. And in this case, we use the nuclear L for channel 1 as main object. So first, we recognize the all nuclei, and we use the intensity module for channel 2. The channel 2 is the LC3 EGFP, the green signal, as a object 1. And as I said before, nuclear L is nuclear L recognize the individual nucleus, and intensity module recognize the various objects like organelles. So the intensity also can recognize the nucleus, but there have a lot of parameter to recognition. So it is more complex. The intensity module is difficult to use, but so if you can set if you can adjust appropriately, so you can recognize the various object like organelles. So in this case, we use the intensity module to recognize the LC3 puncture. Okay. And first, we will adjust the nuclear L. And there are a few parameters. And first, we can see the preview and check the results quickly. In this case, the default setting is okay. So each cell can separate it. Each cell can separate. So we can separate in each cell, and there is no misrecognition. Okay. Yeah, it's done. And then we will next set the intensity module. So in this module, so we want to recognize the LC3 puncta as a spot. Okay. And yeah, there is a lot of parameters to recognition. And we will set the 3 part mainly. First, the threshold setting and segmentation setting, and modify objects. Okay. First, so we check the preview on the default condition and check the recognized results. So it's very small. But yeah, in this case, we need more separation, and we set the threshold level to 400. So, and we can in Novicite, so you can see the intensity information in sorry, this part. Okay, so this is the intensity information, and we can set the threshold level manually, and the reduction is the 100%. The reduction is high reduction is the high resolution that recognition to in high resolution. How should I say? So you can recognize the very with high resolution. Okay, so in this case, so we want to recognize each LC3 spot. So we set the reduction to 100% to recognize small spot. Okay, and we will check again. So we can use the preview area, limit preview area, to focus on this area. So we use this function. And click on preview again, and you can see the detailed area. And it's better, but need more separation, I think. So because this is recognized as a single spot. But I think that there are a lot of spot that we need more separation, and we will so we will set the setting for separation. So the check region setting and set the object size to 2 pixels. And click on preview again, and we can see can you see that? So each spot are separated. It's better, I think. But there are the very small particle, like debris, like a noise, something like that. So I want to remove these objects. So we use the filter function, this function. So exclude the object with the size of less than the 10 cubic pixels. Okay, we can try it. and check them. Can you see that? So they're the very small particle, but after the using filter function, so the object like debris has been removed. Yeah, it is get better. Okay, this is the object setting, object recognition. Oh, okay. It's moved. It is the main part in this video, and we so in this case the parents is main object and the child is object one. But we want to change this relationship. So as I said before, so this analysis starting from nucleus, but the LC3 puncture are localized in cytoplasm. So the lesion of the nucleus should be expand. Okay, so we can adjust this relationship in the object mask. And so actually, so this parameter is quite confusing. It is complex. So I want to show the result. So we can set the width and distance to adjust the relationship between two samples. In this case, we're starting from the nucleus and we expand the region to the cytoplasm. Like this, okay? Yeah, based on the nucleus as a starting point, and the region of cytoplasm could be recognized, okay? So we can use the LC3 puncture in cytoplasm. Okay, then, so we also set the parameter, and in this case, so we choose the object one counts for main object. Main object is the nucleus. So we will classify. So we will do the classification in next step. So we will use we want to use the number of object one in each nucleus, right? So yeah, we choose these parameters, and we will do the run to analysis, and so we set the object one counts as x and y axis. And if you set the same axis, the same parameters as the x and y axis, so we will Nobisai show the histogram like this. And you can also check the plot and load the image, the corresponding image in the viewer, and you can check that. And we create the region. And so so I said before, as I said before, the more than 3 LC3 puncture is autophagic positive cell. So I set this, and so in Novisite, so we can change the x point, this point manually. So we will change the X point to the 2.5 to 200. So this means the, oh, please wait, please wait just a few minutes, sorry. My battery is so low.
Oops.
Oops. Sorry, please wait the few seconds. Sorry. Okay. Okay. Can you see that? Can you see me and can you see my video?
Yeah, we can see your
Okay.
video and we can hear.
Okay. Okay. I'm so sorry. Okay. Go ahead. And so this is a classification, and then so we will set the gate. Yeah, we can also check the object gallery. And so we will create the gate like this. And in this case, so we set the gate name and definition. The autophagic cells is the R01. R01 is more than 2.5, more than 3 to 200. Okay. This is the SC3, the for autophagy positive cell. Okay. And next, we also set the normal cell. So click on new button and set the name and definition using Boolean expression. The normal cells is not R01, the less than 3 SC3 punct. Okay. And we also check the in gallery function. And left panel is autophagy positive cell, and right panel is the normal cells. And if you use the gallery function, you can see the result of classification. And you can check the classification results side by side using gallery function. The left is autophagic cells and right is normal cells. Okay, and you can also see the table, the result on table. And in this case, the control sample has the 1% positive cell, autophagic positive cell, but the treated chloroquine, the sample treated chloroquine, the 83% cell are positive. Okay, so this is the result, and we can also see this result on heat map like this. So if you have a lot of sample, so you don't want to see the result in table, so you can see the result on heat map, the very graphically. So you can see the E3. Okay, and then so you can also see the check the result in the viewer. And this is 3D viewer. The red object is the autophagic cell, or autophagic cells. And you can check the another sample. So this is a control sample. There are a few autophagic cell. You can see that. And you can download this data, and you can make the graph to show the which sample is the autophagic cell, autophagic cell ratio, and you can make a graph to show them. Okay, that's all for this demonstration. And if you have any question about this demonstration video or the previous session, I want to answer them.
There are questions in the chat box, but participants, if you have any questions, you can unmute yourself and ask.
Mhm. No question? Okay. Yeah, if you have any question, so you can ask me or you can ask the Ganesh, and the email address of Ganesh or in the chat box. Okay. Okay. I want to finish my presentation. So I want to show you the combination of Novicide and confocal systems and tissue clearing technology can be used to analyze cell models in more detail. So we also believe that the Novicide or FB3000 confocal system can be used for drug screening or and various researches. So we would like to contribute to. So we would like to continue to contribute to various research fields. Yeah, thank you so much. And this work was supported by these members. I'd like to acknowledge them. Thank you so much. Thank you for listening.
Thank you, Ishihara-san, for that wonderful talk. And thank you for explaining 3D segmentation and how we can use Novicide for that. I hope the participants benefited from that. So I'll once again like to thank you. And if there are any further questions, the participants can directly contact Ganesh or Ishihara-san. We have Ganesh has already given his mail ID in the chat box. And now we would like to request everyone to switch on their videos so that we can take a session photographs.
Okay.
Ishihara-san, you can unshare your presentation.
Sorry, could you say again?
You can unshare your presentation so that we can then take the screenshot.
Okay. Mhm. Okay. Mm. Oh,
Yeah,
sorry.
thank you.
Yeah, thank you so much,
Thank you,
everyone.
everyone.
Yeah, thank you, everyone. Bye.
Thank you, Ishihara-san. Thank you very much.
Thank you very much. Hi, Ganesh.
Hi. Thank you.
How
Thanks a lot for
how
your
was
time.
that? Yeah, sure.
Yeah,
How was that?
it was good. Like, so we will take it forward. Like, if somebody is interested, like, we'll be happy to demonstrate the software
Yeah.
with their sample. So they can either send their sample through email. We'll
Mhm.
try to come online and then analyze. Otherwise, like, if at all we are keeping the system somewhere nearby, we can intimate them.
Mhm.
Thank you. Thank you very much.
Yeah, thank you very much. I really
Thank you,
enjoyed
Akshay,
that.
for. Thank you.
Thank you, Dr.
Thank
Ganesh, for
you.
joining us. Thank you.
Thank-
Yes.